It's a coral health and bleaching assessment a color reference card for Hawaiian corals. So throughout my career, I've been extremely fortunate to experience some very vibrant, beautiful, colorful coral reefs. And not only the beauty that the coral reefs provide, they also provide they provide homes and resources for nearly 1/3 of our marine organisms, and also food resources for more than a billion people, even though they only inhabit less than 1% of our oceans. However, coral reefs such as this one that you see here in front of you and across the world are undergoing severe ecological decline. And it's completely devastating what's occurring across our reefs. And it's estimated that this decline on our reefs is happening about twice as fast as rainforest decline. However, we don't see these changes that are occurring underneath our oceans because it's in a concealing medium, it's underwater, we don't live underwater. I wish we did, but we don't. So some of these changes that are occurring are happening at a very fast pace, and it's really unfortunate it's happening. And part of the talk I'll be talking about today is trying to capture the changes that are occurring and raise awareness about what we're seeing on our reefs. So I'm a marine ecologist, and particularly I'm interested in understanding how local and global drivers of environmental change impact our marine ecosystems. So when I talk about drivers of environmental change, I focus on local drivers such as sedimentation, eutrophication, invasive species, and then also global drivers such as warming and acidification. And we're really interested in understanding how environmental change has impacts across biological organizational scales, such as organismal level, community level, and ecosystem level. And the way that we really understand these processes that are happening is we use a combination of lab manipulations and also field-based approaches. And for my research group, we're particularly interested in answering some of these research questions. They're very broad. The first are, what are the impacts of environmental change? What are the mechanisms of resilience? And also, how do we use this information, the science, for solutions? And this is what I'm going to be focusing on today, is an example of how we've used the science to create solutions in the Hawaiian archipelago. So as you may know, coral reefs are built of individual coral colonies. As you see here, a beautiful image of a Hawaiian coral reef. If we zoom in and look at a coral colony, the coral colony itself is made up of thousands of individual coral polyps. And within that coral polyp, within that coral tissue, there are symbiodinium, or the algal symbionts that are associated with corals. Now, zoom in version of that here, that provide up to 90% of the coral's energetic requirements. So the coral and the algae, and I'll be calling them zooxanthellae throughout this talk, have a symbiotic relationship where they both benefit from each other's presence. So the algae has carbon dioxide from the coral through respiration to fuel photosynthesis, and in turn, the algae provides the corals with energy and nutrients and oxygen. So they have this really unique and fragile symbiosis. However, if there are any changes that occur in the environment, such as an increase in temperature, acidification, sedimentation, so on and so forth, we'll be focusing on increase in temperature today. If the water gets too warm, what we'll see is there will be a breakdown in this relationship between the coral animal and the algal symbionts. And this breakdown is called coral bleaching. The reason why we call it coral bleaching is because the algal symbionts do give a lot of the color to the coral that we see. So if we see a breakdown in that algal symbiont and coral animal relationship, we see a loss of the zooxanthellae, or the algal symbionts, and therefore we see a loss of that color. The coral tissue itself is relatively translucent. So what we see is we see through the coral tissue to the carbonate skeleton, and that's that white color that we observe. So working with corals is a really great system to work with because of this relationship between the coral and its zooxanthellae. And we can look at how these animals may be responding across individual levels, to population and community levels, to entire ecosystem or reef scales, to understand what kind of environmental change has on the impacts it has on these unique marine ecosystems. So as you may know, there has been a lot of warming that has occurred in our ocean's waters, and this has in turn pushed corals into their upper thermal limit and pushing them beyond the boundary in which they can no longer maintain this symbiosis with their algal symbionts. So here I am showing you a map, a NOAA Coral Reef Watch map from the last global bleaching event that we saw. This last global bleaching event occurred between 2014 and 2017. It lasted 3 years. And this map is showing that all of the dark color reds are showing high levels of bleaching that occurred in these waters. The red color is likely high levels of bleaching. The oranges is it's warm, but maybe not so much bleaching. And then the blue colors would indicate no stress at all. So even just looking at this map, we see across our oceans, there was no period in time over this 3-year period where no ocean, no coral reefs experienced no stress. And it was estimated that the last global coral bleaching event impacted nearly 70% of all of our coral reefs. And particularly, we'll be focusing about and talking about Hawaii today. So as we zoom in and look at how Hawaii and the waters surrounding it were impacted from these bleaching events, what we saw was that there was warming that occurred in 2014. So the warmer colors came from the north and impacted the islands. And then in 2015, we saw the opposite trend where the warming came from the south and impacted the southern parts of the islands. And then in 2019, it was kind of all over the place, the warming that happened. So essentially these reef ecosystems, the most isolated reef ecosystems in the world, were extremely impacted by the warming that has occurred in our oceans, along with other reefs across the world. So in 2014 and 2015, it was extremely chaotic in the Hawaiian archipelago. For me as a scientist that was really interested in coral stress response and having not seen coral bleaching before, I was really excited. I was like, whoa, this is really interesting to see all of these individual corals of different species in different areas all respond the same way. And that was in 2014, I was excited about it. And then in 2015, I was very depressed and upset about it. But what we were trying to do at this time is we were trying to get as many eyes in the water as possible to capture the amount of bleaching that was occurring so we can get an idea of how bad was it? How many corals and what areas were impacted by these warm water events? And we estimated nearly 50% of the corals in the Hawaiian archipelago were impacted by this 2014-2015 bleaching event. Again, this is a back-to-back bleaching event that occurred. So this is very alarming, but I will mention that if the temperatures do subside and the corals are able to reestablish that relationship with their symbionts, their zooxanthellae, then the corals can recover from this thermal stress. However, if the temperature is too extreme and it occurs for too long, the corals are essentially starving to death because they no longer have that energetic requirements from their zooxanthellae that's supplied from their zooxanthellae. So the corals will starve to death and die. So while we can assess bleaching as a stress response, what we really need to know is how much mortality occurred across the Hawaiian archipelago. So when we pulled all of this information together across different organizations, different scientists, different resource managers, and so on, we wanted to get an idea of how much bleaching occurred, how much mortality occurred, how many corals died from this back-to-back bleaching event. It was estimated that the mean mortality across the main Hawaiian Islands was nearly 34%. So it doesn't sound too bad, but we've lost nearly a third of the reefs in Hawaii. And then you can see here by this figure that based on where we were on the island, there was a lot of variation in how much coral was lost. From Ni'ihau and Kauai all the way over to the left-hand side of the screen, we saw nearly half of the corals were lost in those areas. Versus the Big Island, we saw about 40% decline of those coral reefs in that area. And if you look at Oahu, it was really variable between 7% in some areas up to 85% in the other areas. So a lot of variability in how the reefs were impacted. This is likely due to the warming where it came from in 2014. So it came from the north. So there's more warming that occurred in Kauai and Ni'ihau. And then in 2015, it came from the south. So there's more warming on Big Island and then Maui Nui area. Alongside that, there are a lot of different local factors of environmental change across these Hawaiian Islands. So that likely contributed to the mortality that we saw as well. But the bottom line is we lost nearly a third of our coral reefs on average across the islands. And if we look at how sea surface temperature or the warming has increased over time, here I'm showing you some offshore data of the temperature, which is on the y-axis in Celsius versus time. So this is dating back all the way to January 1956 and how warming has increased. So we see a steady increase in warming in the waters off of Hawaii about 1.2 degrees over the past 50 years. And we've had 4 documented bleaching events in the Hawaiian archipelago, in the main Hawaiian Islands. So in 1996, as shown here, 2014, 2015 that I mentioned before, the back-to-back bleaching, and then we had a more recent one in 2019. And what we're expected to see that these bleaching events, these warm water events, are going to become more severe and more frequent. So it's not looking good. However, what we need to do is we need to equip our resource users with the knowledge that we have as coral biologists to help us understand if a coral is healthy or not. So when I was a graduate student at the University of Hawaii, I was working with my PhD advisor at the time, Dr. Paul Jokiel, who was the first scientist to define the thermal limits of how warm is too warm for corals. And when I was talking with him, he asked me, how do we tell if a coral is healthy? So I proceeded to list all of these biological response variables. Well, we measure this and we measure that and so on and so forth. And he was like, no, you look at it. And this is getting back to the same thing I was mentioning earlier, is that if we do not see the color in the coral, then that relationship between the algae and the coral has broken up and the algae has been lost. And this is what the schematic here is showing. A healthy coral has zooxanthellae, or the symbiotic algae, inside of its tissues. If the water gets too warm, that relationship breaks down, you lose the zooxanthellae, and then you see a white coral or a lighter color coral. So essentially, we needed to equip all of the citizens in Hawaii, visitors, residents, locals that live there, and resource managers with some tool to help them assess the health of the corals across our islands. And if we could do this, we can have this rapid response network where people in the water every day could report if bleaching was happening or there was an unhealthy coral. And then that information can get relayed to resource managers and scientists. And then those scientists and resource managers can deploy a rapid response team to go out, look at the corals, and conduct surveys and monitoring. But we really needed some kind of tool for this. So in Australia, there has been a color card, a coral health color card, the Coral Watch Coral Health Chart that was developed early on in the 2000s, and it was published in 2008, and it has been extremely effective on engaging citizen scientists with monitoring the health of their reefs. And we were thinking, wow, this is brilliant. We can correlate the color of the coral with the health of the coral and give it to people and have them out on the water and report back to us. This has been extremely effective in Australia and the Indo-Pacific waters. However, these colors that we see for the color chart for Australian corals and the Indo-Pacific are very different from the corals that we have in Hawaii. So in the Indo-Pacific, there's thousands of types of coral species, and in Hawaii, we only have between 40 and 60 types of species, and really only 10 of them are really dominant in the Hawaiian Islands. So we really needed to think about how do we have a color chart that it more encapsulates the colors that we would see on the reefs across Hawaii. Because as you see here, if we were to use the color health chart, the coral health chart that was developed by Australia, it's not really representative of these are the dominant species we see in Hawaii. You see we are missing a lot of these purple colors, and some of these yellows are there, but we're missing some of these oranges. So we really needed to revamp this and follow this really successful model that they have there for corals in Hawaii. And this was the overall goal for us to have our own color health chart that would be representative of the colors there. And that's what I'm going to be talking about today, of how we've developed this tool and the science behind it and how we've implemented it across the Hawaiian Islands. And the overall goal for this was not only to provide a color for the citizen scientists, for the resource managers, for the scientists. What we wanted to do is to correlate these colors with an actual physiological health or condition of what the coral was experiencing. And we wanted to make sure that this was representative of the health of the coral through these different methods, so such as the photosynthetic state, the fluorescence, the symbionts, and the chlorophyll that was associated with the symbionts inside of the coral. And I'm going to step through these and how we actually quantified this. So what we first had to do is we had to collect some corals. We decided to go with 6 species of corals that were the most dominant color corals in Hawaii, and also a variation in the colors that we would see. We collected several colonies, and we fragmented them into a lot of different individual coral fragments you see here, different individuals, and had them inside of our flow-through mesocosm facility at the Hawaii Institute of Marine Biology. And then we simulated a bleaching event, so a warm water event. We increased the temperatures, and then with that, every couple of days, we would take a couple of the corals out. We would photograph them, as you see here, so that we can have an image of what the color looks like. And then we'd also do a coral health assessment. And with the coral health assessment, we would look at the photosynthetic efficiency of the symbiosis that was occurring, or the photosynthesis that was occurring by the algae. So we used pulse amplitude modulated fluorometry, or you'll hear me say PAM. We use PAM to get an idea of how efficient photosynthetic processes were occurring. So how efficient was photosystem 2? And then we also sacrificed the coral and then looked at the symbiont density. So how many zooxanthellae were in that coral, and how much chlorophyll did those zooxanthellae have? So this could give us an idea of what the health was, or how present were those zooxanthellae in the chlorophyll inside of it for photosynthesis. And what we saw here was the symbiont density, which is on the y-axis, and the days of exposure. This is the days of exposure to warming. We saw, as we would expect, a general decline of the symbiont density with prolonged exposure to elevated temperatures. We also did the same thing. We saw a similar trend with chlorophyll A concentration. So the longer they were exposed to elevated temperature, we saw a decrease in the amount of chlorophyll that the symbionts had. And you can see that there is a lot of variation in this data. It's kind of messing all over the place, because this was pulling across all of the different species that we looked at, the 6 different species that we looked at. So we see a lot of variation. This is likely because different coral species have a different mechanism for dealing with stress. Some may decrease the amount of zooxanthellae it have, some may decrease the amount of chlorophyll within those zooxanthellae. So there's different things happening here that we still need to dive into. But what we saw was, in terms of the fluorometry, or what was happening with the PAM measurements, this gives us an idea of what the overall photosynthetic efficiency was. We saw with increase in exposure to temperature, there was an overall decline. That was a very nice fit across all species. You increase the temperature, you see a decline in the photosynthetic capacity of the algae within the coral. And we also saw that the quantum yield, or the PAM fluorometry measurements, correlated really well with the colors associated with that too. So this was our key factor that we saw was nicely correlated across all species. So along with doing the coral health assessment that you see on the right-hand side, we also need to process all the photographs from the corals that we took. We took photographs of about 533 corals, and we then had a resulting, I think it was tens or hundreds of thousands of different color palettes associated with those corals. Within each of the corals, we selected 10 random spots on the coral that was illuminated very well, didn't have any shadowing or occur, and those are the results that you see down here, some of the screen captures of that. And we went through these and tried to capture the most representative colors of these different coral species at that point in time. And then together with the coral health assessment and the photographs, the colors that we had from the photographs, we then began to start thinking about how are we going to put all of this information into a user-friendly card. And this was actually the really difficult part, because as a scientist, we're thinking a logical step of this, this, and this. But when we were talking about developing the card, we really had to think about it from the end user side of things. How do we revamp this and provide an easy way for a citizen to be able to use this? So ideally, beginning, we thought, well, we could have these gradient of colors. That would be an ideal for us as scientists to have an idea of the most differences that are occurring on our reefs. And then we decided that we actually needed to break it up and have distinct colors associated with the coral. And then we saw that within this version of the Koʻa card, we had 4 whites, and we don't need 4 whites because white is bleached and not healthy. So what we ended up doing is revamping this 3 times and getting feedback from the community to make it as efficient as possible. So this is what our end result looked like. And ideally with this, we saw, we have 4 different quadrants that I'll be talking about shortly, but we go from a darker color, so there's more pigment, more zooxanthellae, higher levels of chlorophyll, to not so pigmented, so less pigmented. as you work your way around the wheel. So 27 would be an indication of a healthier coral versus 19 would be a bleached or stressed coral. And this ended up being a more effective way to capture the changes that were occurring for the corals in Hawaii. So then after we had this first round of the development, what we needed to do is really validate that the colors that we chose and the health parameters that we measured were actually representative of another bleaching, representative of what we saw in the field, and also if we were to repeat this experiment to make sure that they were valid. So we did card validation, which I'm going to call phase 2. We looked at doing the lab validation, a field validation, and observer variation or validation. So with the lab validation, what we did is essentially repeat this entire process all over again. Since we had so much fun doing it the first time, we did it all over again, but this time we only had 108 individuals. So we just did less replicates within each of the species, and then went through the entire process again, except for the card development. And then what we saw with that is we wanted to get an idea of, are the colors that we're measuring are actually the colors that we saw? And also, do the color health parameters actually match up? So for this next slide, I just want to orient you to the Koʻa Card. And like I mentioned, we have 4 different quadrants, and these quadrants were separated by colors that are more representative of the species in those areas. So we might see these types of species will be in this quadrant versus some of these species might be in this quadrant. And then these are the redder colors or representative of different species. Sorry about that. And then this quadrant number 4, is more the purple hues associated with those. Because if we put these colors all together, it would be really confusing and not very effective for the user to be able to turn them and figure out what they're looking at. So we were hoping by separating them by the main dominant color hues, it'd be easier for the end users to use them. And throughout this entire process, me as a trained biologist, as a scientist, it was really interesting to go to the color side of things and the design side of things to really put on another hat and see what's really more effective. And it might make more sense to you as a scientist to have it a certain way, but when we had feedback from the end users, the people who would be using it, it wasn't as effective. So it was very interesting going through this whole process and just learning from going from the end and working your way up. So for this next graph and the one after this, I want to orient you to these figures. So I have the 4 different quadrants that I just mentioned. So 1, 2, 3, and 4. And then on the y-axis, I have the different colors of the color scale. So this is red, green, blue color scale for all of those. And on the bottom is the different color card colors that we chose for the color wheel. These bars that are in each of these colors, you'll see that 0 has no colors, of course, because it's no colors there. But these are the color numbers that are associated with the colors on the color wheel. And what we wanted to do is have an idea of if the color wheel values are a certain number, so like 245 for this color for red, and this color 220 for green, and then 160 for blue, we wanted to make sure that the color images that we got from the corals actually matched up with that. So what we did during the second round is we then took the numbers from the colors that we called this coral number 11. We wanted to make sure that the colors then aligned with that. And while we do see some of these don't necessarily agree, we do see overall there is the majority of the colors do agree with the colors that we saw within the corals. So that was relatively exciting for us because different people see different colors. So we wanted to make sure that there was agreement between what the camera was reading and what the card was reading, so those can match up without having any observer bias as us as humans. And then on the other side, we wanted to make sure that there was alignment with the measurements, the biological measurements that we took of these corals. So here I'm showing you the quantum yield. So this is the measurements from the pulse amplitude modulated fluorometer. So this is the photosynthetic capacity of the corals, how well photosystem 2 is working. And the open circles are phase 1 for each of the different colors. So if we called a coral number 6, this is this overall measurement for the quantum yield, the FE over FM. And then when we looked at phase 2, again, we had a subset of corals, so we didn't have all of the colors represented, but we saw that overall there was general agreement between the measurements that we took, and we classified it as this color had similar colors between the first phase and the second phase. And we also did this with the other measurements, the chlorophyll and the zooxanthellae as well. But again, we saw the highest correlation between color and FV over FM, the photosynthetic efficiency. So I wanted to share that with you, but we do have all of those other measurements in the manuscript in the paper. So after the lab validation came the fun part. We got to go into the field with the Koʻa card and have an idea of were we seeing the colors actually represented on in the water and in the field. So here on this map, I have the blue sites are where we collected the coral, so mostly on the eastern shore, and that was due to how accessible these reefs were for us. And then the red is indications of where we did some validation of those. So we did 11 different sites, and the depths of these sites were between almost 1 meter and 2 meters. And what we saw was that generally all of the corals were, the colors were represented. We do see that some of the lighter colors were not observed in the field, and that's likely because there wasn't a bleaching event occurring at that time. We did this in the winter time, so we didn't have those warmer waters, so we didn't see that bleaching response happening. And then also, I think what's most important is that we only used 6 species of corals to develop this card and to develop these colors, but we found that these colors were representative of 17 coral species in Hawaii. And as I mentioned before, there's only about a dozen corals that are really dominant in the Hawaiian waters. So this was really great news for us, that even though we only use 6 coral species, we also saw that this matched really well with a lot of other coral species as well. And then another thing that we didn't really consider as scientists, but it was really important to think about, was the material that the Koʻa card was made out of. How was that going to hold up? Since color was so important in order to tell the health of the coral, we needed to make sure that that color was going to hold up through use and abuse of the card. So we did some exposure experiments with the card itself, and we used 2 different types of material. The first version of the Koʻa card, you can see here on the bottom, was made of a non-biodegradable material. And then the revamped version, the final version of the Koʻa card, was actually made with a biodegradable material, because we thought that we don't want to contribute to the plastic problem. We want to be part of the solution. So if someone did lose a card, we wanted to be able to be biodegradable and not stay forever within our oceans. So what we did is we did exposure to the sun, left it out in the sun for a few weeks. We kept it underwater for a few weeks, and then we scratched it to try to see if any of the color would come off. And what we saw overall was the biodegradable material, even though it's biodegradable, it held up and it held the color longer than the other material that we were using. So that was really great news for us, just a side note on that. And the last validation that we wanted to do was observer-based. So as we all know, as humans, we all see color slightly differently. So if we are not able to distinguish color within ourselves between me as an observer and another observer, there might be some variation with the colors that are reported for the Koʻa card. So we wanted to do some studies to get an idea of what was the overall precision and accuracy, well, I guess more precision of what the colors were called based on the observers we had in Hawaii. So we had numerous volunteers to help us out with this study. We recorded their age, gender, how familiar they were with corals, their education, and to get some feedback on how easy the card was to use as well, so we can make adjustments from there. And overall, what we saw was that there was no real or clear difference between the age, the gender, and the coral familiarity or the education. Everyone was essentially calling the coral the same number. So here on this figure, I have what the mode was of what they called coral number 1, and it was very similar with the mean and so on and so forth, but there wasn't any stark differences between the age or the gender explaining that variation that we saw. We did notice, but it wasn't significant, that majority of our testers, our volunteers, I believe it was 80% of them were male, and we did see that the men were seeing color slightly differently, but it wasn't significant enough that it would cause any deviation in how they would call the colors. And then we did see higher precision with those that were more familiar with corals. So from this study, we determined that we needed to have some training materials that came alongside this Koʻa card. So everyone had a little bit more training in corals, how to identify them, and also how to use the card appropriately so we can help reduce some of that variation that is due to human error. So that leads into my last section here, which is the implementation of the Koʻa card. And again, remember, this is scientists developing this card. We really needed to get a lot of feedback and help from people outside of our area with overall implementation. A lot of things made sense to us. We're like, this is easy. This is how you do surveys. But it wasn't necessarily as intuitive for some of our end users. So with the design of the back of the card, it also went through a lot of iterations. But what we decided was that we needed to provide the people that were using this card with some choices or some options of exactly how to do a survey. So we had 2 different survey types that we recommended, and then we also needed to provide some information for the end user to know the different coral shapes, because if we pair the coral color with the coral shape, then we can get an idea of what the coral species is for us as scientists. Again, there's not that many species in Hawaii, so it's a little easier for us to narrow down what they're likely seeing. And then we needed to give them some information of how to characterize the coral. Should they be looking at the entire color? Should they look at the lightest and darkest? So we wanted to have that all standardized. And then also we have an entire reporting platform as well. So we made some videos. This is available on our website that go over the different surveying methods, and also an entire training video. So this ended up being a lot more than we previously thought. We just thought we could develop a color card and just give it to people, but we needed to make sure that they were using it effectively. So we went over exactly how to do a transect with them and provided a video of that. And this would give us an idea of what area they were surveying, and if they were not biasing their surveys to only bleached corals. We wanted to make sure that they were capturing corals that were healthy in an area and corals that were bleached, of course, but we wanted to make sure we weren't just capturing corals that were unhealthy as well. And then we provided another method, which was a haphazard method, in case the surveyor, the end user, didn't have a transect or a line or some way to measure the area that they were doing. We provided another method that they could use, which we considered haphazardly, just selecting 20 corals and then randomly, and then looking at the color of those corals and recording their shape, their color, and then also providing any additional information that would be helpful for us, where they did the surveys, how deep were they, what was the time, and if they could, what was the water temperature for that. And then also with that, we needed to have an idea of what the shape of the coral was, and that could give us some indication of what coral species that they were looking at. So we went with 4 broad shape categories because we didn't want to provide too many options and it would be too overwhelming for our end users. And these seem to be very great categories to classify our different types of coral species into, looking at branch corals, mounding corals, crest corals, and plate corals. That were really well representative of the coral species they would likely find. And then lastly, we wanted to make sure that they had an idea of how they were going to assess the color of the coral. And we talked about doing the light and the darkest part of the coral. And then that provided more information, and it added more variation to potentially how the surveyor was collecting that information. So what we decided was to pick the best color that represents the entire colony. So while you might see some parts of this coral here has some bite marks from a fish right here, but overall, this is a bite mark and not necessarily bleaching. We want to know what the overall state and the color is of that coral, so that they could report that color. And then lastly, we wanted to make sure that we had some kind of data template for them to collect all this information. So we provided some data sheets for our end users that could take out into the field, record this information, and then report it back to us. So with all of this, the implementation side of things, it became really important and really valuable to us to have somewhere for our end users to report all this information. Originally, we were thinking we could just send out a Google sheet and they report all this information, but it was getting so large and the response that we got from our end users was so great. We needed some help. So fortunately for us, the Pacific Island Ocean Observing System, PAC-IWIS, was very friendly, really nice, and they supported our project and hosted it on their website. And even on their website now, you can see an overall view of what the problem is, what are coral bleaching events, how to participate, how to get a card. Also, I should mention that we had a donor print all of these cards for us. I think he printed 5,000 cards that we just gave to people. We didn't ask people for money. We wanted more people in the water. We just gave these cards out, and we also dropped them off at the local resource managers agencies. And then it went through how to submit your observations as well on their website. So we were really fortunate to have them be a part of this cause. And on the PAC-OS website, the data entry form is very, very simple, where the end user can provide the color, the coral color and the coral shape. That's essentially all we need to know, but the more information they provided, the better. So if they knew the coral species, then they could provide that coral species. Maybe they have the training, maybe they are more familiar with corals. They can give us an overall assessment of the area that they surveyed and how much bleaching occurred in that area, what was their depth, their time, the date. And also what I think was most important is we wanted to get an accurate representation of where they did these surveys. And not everyone has a GPS with them, of course. I don't even carry around a GPS, but you do have it on your phone, and they can also report this on their phone. But what they ended up doing is they had this interactive map where you can draw, zoom in, and then click, drop a pin where you did your surveys, and that would give us a better idea of where these observations were taken up by our citizen scientists. So together, then we can look at the overall data visualization of how much Koʻa Card observations were reported. And we can do a search term looking, and anyone can do this. We can look by year, so how many observations were made in each year. And that can give us an idea if it was a warmer year, where did they report things versus a cooler year, where were some reports made. Then we can also search by different colors. So if we're trying to look for bleaching, which is the number 0, we can see where bleaching was observed across our different Hawaiian islands. And then also we could look at the different shapes that there might be, if there was a photo provided, and where this information was collected. And like I said, where bleaching was and the water temperature of the islands. So this was a really nice way for us as scientists to visualize it, also resource managers to visualize it, and the end users, where they see their data is actually making a difference and it's recorded, and people can check on this information in near real time. You can go out and do a survey, sit on the beach, and just enter all of this information. So it was really, really valuable for us as scientists to have an idea of where the data was being reported and how it could be used. And then when we zoom in on these different data points, we can get an idea of where these reports were occurring and the different colors of the corals that we saw there. So all of the colors you see there, or they saw a crust coral, it was number 17, so that's relatively dark in pigment, so that one's doing pretty good. And we can see the date and the time that observation was made. And then also some other users provided a photo with the Koʻa card, so we can have an idea of, oh, that coral looks pretty good. This is a color card for reference. And they also put their dive computer in there with the depth and the temperature as well. So all of this information hosted at one site that is easily accessible by scientists, resource managers, and the public as well. So this is really excited to see all of this come together. And I should mention that this was like an 8-year project, really 4 years together, and multiple people trying to pull all of this information and get this off the ground and lifted. So when we go back to this overall assessment and how we can better monitor our reefs and indicate where bleaching is happening and respond to that bleaching and have some mediation strategies surrounding that to help corals recover from these bleaching events, the Koʻa Card has been a really effective tool to equip our citizen scientists, our resource managers, scientists, so on and so forth, with some tool that provides a standardization across all these different groups to have an overall assessment of how healthy of a coral is, report that, and then we can deploy resource managers, scientists, and a response team to follow up on that, or to help make mitigation strategies to relieve some of that pressure that these corals might be experiencing. So like I said, this has been a 4-year project. It's actually all started with my PhD advisor, who said, just look at a coral and you can tell how healthy it is. But we really needed some way to give a tool to our end users so they could tell how healthy it was, because not everyone has 40 years of experience looking at corals. This Koʻa Card for Hawaiian reefs gives those end users, those public, a tool that gives them almost 40 years of experience of looking at corals. And it wouldn't have been possible without him suggesting developing a card and equipping our public with that. But then also the generous support of our sponsors for this project as well. We received partial funding for this project through the Council Foundation and from the Hawaii Community Foundation. And then also we had a lot of support from the Hawaii Division of Aquatic Resources, or Hawaii DAR. And like I mentioned earlier, these cards were printed for free, so we could give them to the public for free, get more eyes on the water by content. It was really great that they provided this tool for us, because originally this card would cost almost $10 a piece to print those. And like I said, this project was supported by the Pacific Island Ocean Observing System, and this was in collaborations with myself and my colleagues at the Hawaii Institute of Marine Biology in the Coral Reef Assessment and Monitoring Program. So I'm really excited that it's come to fruition. It's completely done, and now it's released out into the world. And as you saw, a lot of people are using it. And hopefully, next time we have a bleaching event, we can have more eyes in the water and a tool for those eyes that are in the water to accurately assess the health of our corals. So with that, I am happy to take any questions. I think we have plenty of time for questions. And I think I saw in the chat there was a good amount of questions for me. And thank you all for listening.
Yes, thank you very much for a very informative presentation. Yeah, I will start by asking few questions that I took from other people who could not attend and will be watching the recording later on. My first question to you is that you indicated in your paper that the Australian health card has been used for coral reef assessment in Kenya and Iran. Have the Hawaiian Koʻa Card been used in any other coral reef assessment anywhere in the world?
No, so this card is dedicated for corals in Hawaii. Like I mentioned earlier, the corals in Hawaii are relatively specific to that area because of the isolation of the Hawaiian archipelago. So some of the corals that we made this card for are endemics. They're only found there in Hawaii. This card might be helpful in other systems. One of the recommendations that I have is that since the coral species that you find in different areas are different, the colors for those coral species should be represented for those areas. So if we were to take an endemic species of Hawaii and have a color associated with that, and then try to employ that in the Indo-Pacific, we're not going to find that color, that coral. So that color might be slightly different in the measurements that come from that color and won't be appropriate for those corals. So short answer, it has not been implemented and it will likely just stay in Hawaii. But now even here, I'm in Texas now, we're working with the aquarium here in Texas to create a color chart for the corals that they have here. So they have an idea of how the color is correlated with the specific species they have here. So they can use that tool. So essentially this is like a platform for moving forward to develop in other areas.
Okay, thank you very much. And I have another question before I go to the chat. Related to your study, you actually look at coral occurring at a depth up to 4.5 meter. Am I correct? And what happens if you look at coral that occur below 4.5 meter? Do you think this chart will still be representative?
That's a great question. So we did do up to 2 meters. The validations, what we see is, as we know, as you decrease your depth in the water, you lose a lot of that light. So with that light, you're losing the colors that you see. So this isn't recommended to use at scuba diving depths. We saw about 20 feet is like the maximum that we're still getting those same colors for the corals. So this might not be more representative of the deeper corals. But if you do use this card and you're at depth, we recommend taking a flashlight just to have some more light associated or shined on the coral. But do know that even as you dive deeper, the colors that you're seeing on the card and the colors that you're seeing underwater should be very similar, but again, you're losing a lot of that color. So this is a better tool for shallower coral reefs that will most likely be more impacted from the warming that we see.
Okay, thank you very much. Let me go to the chat and see the question that we have. I think one of the first questions was related to how many volunteers did you use in the whole process?
In total, that's a great question. I haven't sat down and counted the number of hours or the number of volunteers, because we had volunteers associated with doing the actual experiment and then the measurements of counting the zooxanthellae and doing the chlorophyll measurements. We had probably about 20 volunteers just for that aspect. And then for the survey and the observer variation, we had, I believe it was over 50 volunteers total do the surveys for us. At the Hawaii Institute of Marine Biology, we had tour groups at the time before COVID, which I feel like we're all kind of like, before COVID, what's that? We had groups come through, and a part of the tour, we would ask them to take these surveys and to write down, do a test essentially of matching color with coral. So we probably had over 50, but I need to actually go through and quantify how many people we had for the volunteer side of things. And then we also did it with dive clubs to look at the depth difference, but we had most of them, they saw that the color was lost. They used a flashlight, but it was better at shallower depth. So they did more measurements at 20 feet versus at 50 feet for us.
Okay, thank you very much. And the next question, what is your approach, or what approach did you use when your phi coral did not match with standard color card?
That's a great question. So obviously we have 34 colors because there's a white on the card. I guess we have 35 colors and then the white is 0. So we have 35 colors, and all these 35 colors can't be completely representative of the corals that we see there. What we try to do is the most similar coral to that, but then we also took photos of those corals that don't match very well. And from that, we were trying to assess, like, okay, is this more of a cryptic coral species, or how likely are these corals, are these end users likely to encounter this coral? So most of the time we saw cryptic corals that weren't so abundant on the reef wouldn't be captured by this card. Another thing is, on some of the islands, there's different corals that are more abundant. So on Big Island, there's more blue corals, and we don't even have any blue on this card. And that was just because we just don't have enough space. We could make these smaller, but then it wouldn't be enough space for our users to utilize them. So that was one of the biggest criticisms we had, was that we were missing some of these really large colonies that were on Big Island that weren't captured on our card. And our solution, we were thinking maybe we can have another swatch developed for those corals, and then we make a smaller card. We were talking about circles within circles, but we just got to the end of it. We're like, we just need to make it simple so people are using it. So we do acknowledge that not all the corals would be captured with this wheel.
Okay, thank you very much. And the next question, what is the optimum temperature for corals?
Oh, great question. And I didn't mention that. So corals throughout the world are living within 1 to 2 degrees Celsius of their upper lethal temperature. So in Hawaii, their upper lethal temperature is about 30 to 31 degrees Celsius. And the average temperature is about 28. So if we get higher temperatures than 28, they start getting stressed out. And then around 30, we start to see that they're losing their symbionts. And the longer we have those elevated temperatures, the longer it's above 30 for these corals, then the higher levels of bleaching that we see. So even on this card, you'll see that, of course, white is the bleached coral, but you'll see that they lose pigmentation because they're losing their zooxanthellae. So we wanted to capture that instead of having a normal color, a healthy color, and completely bleached. We wanted to get an idea of, is bleaching going to occur? Are they stressed versus they're completely stressed and we all know it, kind of thing. We wanted to have an idea of the gradation to bleaching and from bleaching. So going to bleach and then recovery from bleaching as well.
Okay, thank you very much. The next question is, for how long can they sustain high temperature?
Yeah, that's a great question too. So it depends on how elevated the temperature is. So some corals can withstand higher temperatures for short durations, and some can withstand not as high temperatures for longer durations. But if you have a combination of both, you're gonna see bleaching occur faster. So in Hawaii, we usually see that after about 10 days of over 30 degrees Celsius, we start seeing bleaching occur, but some species that happens a lot faster for those. And also I should mention too, when these bleaching events happen, we have elevated temperature warming waters that also drive this bleaching. But at the same time, if the water is really calm and the sun is out, that interaction between those highlight light levels and also the warming can interact and be synergistic and cause more bleaching to occur. So that's something to be, that's a big concern for us as well. If we have a really nice sunny day and the water's already really warm, the corals are going to be even more stressed out.
Okay, let's move to the next question. Can remote sensing be used in this coral reef assessment based on satellite image only? And if possible, without doing the field survey? I'm just trying to read and rephrase the question in my head.
Yeah, and I see it on my side too. So short answer, that's the ideal world, yes. And that's something that we're kind of working towards. If we have an idea of the spectral qualities of what consists of a healthy coral based on the color, we can potentially use satellite images to capture that. The only limitations that we have with satellite imagery is that cloud cover and also penetration across the roughness of the surface. So we're trying to be able to scale from in-water measurements of reflectance using hyperspectral measurements of the coral and the reflectance of them, and then go up from there. So that is the, what is it called? Like, that's the pipe dream that we're moving towards right now. And hopefully we start moving in that direction, because if we can have larger scales of what's happening on the reefs, and there's some work being done right now to provide proof of concept, then this would be a better way for us to assess the bleaching that's occurring and the health of the reefs. But also having citizens on the ground in the water. Most of these people are in the water every day, and they even name the corals, which is great. So they go and monitor the corals every day. They can provide ground truthing for that satellite imagery that's occurring too. So I think a combination of both is really, really helpful.
Okay, thank you for your response. And I think the last question, once you have the evaluation of coral health, what action can be taken to mitigate bleaching event?
That's a really great question too. And that's kind of like what we're getting down to, is when we see these bleaching events happening, we feel really hopeless with them, because it's just, well, first it was really exciting for me to see, but then after nothing happened and it keeps happening again and again, it's devastating, because you're essentially watching corals die. What we're hoping to do is, by capturing where there's severe bleaching happening and perhaps no bleaching happening, we're trying to see if there's some drivers, the environmental drivers, that are causing these differences that we see. Potentially, there might be some things that are coming from land, such as nutrient runoff or sedimentation, that might be exacerbating the bleaching of these corals, or they might be mitigating the bleaching. So we want to get a better idea if there's anything that we can do locally to reduce that stress that corals are experiencing. And if we can relieve some of those stressors, then the corals can resist, or if they're not able to resist the warming, they can at least recover from the warming that happens. So together with all of this information, we're hoping that this will be informative for not only the Hawaii Resource Management Agency or the Division of Aquatic Resources, but we're hoping that it can be informative for other areas as well. So if we can have an idea of where bleaching is occurring, not occurring, and find out why, what are the differences between these two different areas, potentially we can have management actions that occur. Those can be designing or implementing MPAs, or doing watershed restoration so that there's less land pollutants coming into the waters, or potentially other actions to help mitigate some of that. So that is our goal, our applied management, adaptive management goal, to provide. First, we need to know this information, and then we need to start talking about, okay, what actions need to be implemented to help the corals recover from the stress that they're experiencing. Because unfortunately, the warming is going to continue. It's going to keep going. It's going to get more severe, and it's going to be prolonged, and the corals are going to have to experience that. We're just hoping that we can relieve some of those other stressors so they can keep up with the warming that's happening.
Okay, it looked like I got another question. For large-scale coral reef monitoring, should we move with remote sensing technology? What is your suggestion in this regard?
I think remote sensing technology has really great applications for this type of work. And I think, like I said earlier, there's some limitations with remote sensing, but we're hoping that if we can overcome some of those limitations, this could be a really great tool for us to get a better idea of the health of our reefs, and then employ management strategies for that. So I am a strong advocate for remote sensing technology, because we're limited in the capacity of how many people we can get on the reef and how much reporting that we can do. And if we can have another more sophisticated technology do those measurements for us, that's amazing, that's great. And then we can focus on those mitigation strategies to help corals recover. So I think that we just need to we need to overcome some of the limitations right now. And then with that, provide some more data, some ground truthing, for example, with these Koʻa cards or with in-water surveys. And then hopefully as we move forward, hopefully fast, we'll be able to have a better assessment of what's happening on our reefs.
Okay, thank you very much for your response. I think that was the last question. And thank you very much for accepting to present during this SN Applied Science webinar series. It was a pleasure to have you on, and I hope all of our participants actually learned something from your presentation. And I want to thank everybody who actually took time off to attend our webinar. And you can follow us on Twitter. You can check our website for updates about our journal. And if you have any question, please don't hesitate to contact me about our journal. And if you have any question about this presentation, because we recorded it, we will actually make it available for other people who could not attend. You can contact me or you can contact Dr. Bahr directly with your question, and she will be more than happy to answer. Thank you very much, and have a nice evening, morning, afternoon, depending on which part of the world you're watching us from. And see you next time.
Thank you.
Thank you
Nice
all.
talk.
Yeah.
See you.
Okay, bye-bye.
Thank you so much.
Bye-bye.
Thank you. Bye.
Thank you. Very nice
Bye
talk.
bye.
Yeah, bye-bye.